and welcome everyone to this podcast on cell signaling. Over the next several classes, we're going to talk about various aspects of cell signaling. In this podcast, we're going to talk about the general principles of cell signaling. And this will actually probably take 1 to 2 podcasts to cover all of the material. In the next podcast, or group of podcasts, we'll talk about G protein coupled receptors. We'll introduce them in these first podcasts, but we'll go into much greater detail in the next group of podcasts for the next class. And then we'll finish this group of podcasts on cell signaling talking about enzyme coupled receptors. But let's get started with some of the general principles of cell signaling. We could think of a free cell. It could be a bacterium, it could be a yeast cell. But this free cell has several things it needs to do. It needs to survive. It needs to divide. It may need to differentiate or just change its overall morphology. Or it may need to move. All of that's going to cause a change in the cell shape. Or it may need to move, where it slowly moves in one direction. And lastly, at some point it has to die, where it may go through some kind of cell death pathway or some other pathway that leads the cell to die. Now, a multicellular organism could be a human, some other animal. It could be, of course, a tree or some other plant. Some kind of multicellular organism. Each one of their cells in this multicellular organism has these same criteria, as well as others. I didn't list them all, but they all have to know how to survive, divide, how to change shape, how to grow during, say, embryonic development or throughout their lives. They also have to know when to undergo apoptosis and die. But they're more complicated. There's more signals from the environment they have to respond to. And they have to differentiate between these signals. So these whole organisms are constantly hit with many different kinds of signals to do various things, but each one of these cells has to be able to differentiate between these signals so that that particular cell's desired outcome is achieved. So now let's think about the process that these cells are going to respond to external stimuli. And we're going to focus on the process right now of signal transduction. There's other ways the cells can communicate with the environment, but this is going to be the focus of our discussion. And signal transduction is a process by which cells communicate with each other through external signals. And so let's label a basic pathway here. So you're going to have this signaling cell, and it's going to produce this signal. And this signal, so let's label that signal, will move to a target cell. And that target cell may have a receptor on the outside of the cell. We'll talk later how these receptors can be on the inside, but we're just talking about a general pathway right now. So it's going to bind to the outside of the cell. When it binds to the outside of that cell, a signal will happen, and maybe there's a protein inside the cell that will respond to that signal. So right here, let's label this as an intracellular signal. This protein here that is modified because of this intracellular signal may go on and do something very specific, or it may go into the nucleus and turn on specific genes, depending on which kind of signal and which kind of receptor we're talking about here. But the end result is that through this signaling pathway, we get some kind of a response. Maybe it's a response for the cell to divide, maybe it's a response for the cell to move, or maybe it's a response to have the cell die. This signaling molecule, I'm going to spend a little time talking about that. This signaling molecule can be one of several different molecules. It can be a protein, it can be a peptide, which is a small protein, it could be an amino acid, it could be a nucleotide, a steroid, it could be a fatty acid, or a lipid. It could also be a dissolved gas. There are thousands of possible signals. But regardless of the signal type, there are really 4 communication pathways that these many different signals could enter. And so I want to spend some time talking about these 4 different pathways. So we're going to talk about 4 communication pathways. I'll list them first, then we'll talk about them. Endocrine, paracrine, synaptic, and contact dependent. So let's start with the endocrine pathway. The endocrine pathway is a long distance signaling pathway using hormones made by endocrine cells. The signal that is made through the endocrine pathway is more global, meaning that as it's being made and it enters the circulatory system, it can go to any part of the body. So let's put that last part here, travels via circulatory system. So let's draw this out here. Here's our endocrine cell. As an example, this could be a beta cell in the pancreas. It's going to make a signal, and that signal is going to enter directly into the circulatory system. And if it's a beta cell from the pancreas, it's quite likely it's going to be insulin. And it's going to go throughout the body and interact with various target cells. So for instance, it might interact with a muscle cell, where it will leave the circulatory system and bind to a receptor of this muscle cell, which will lead to a response. And if this were to be insulin, it would lead to the response of glucose moving into the cell. So endocrine systems, the key features is that they're made by specialized cells, the signals are, and they travel throughout the body of the organism and can interact with several different targets. Next, let's talk about paracrine pathway. It's a little opposite the endocrine in some ways, in that it's more of a local signaling pathway. That communicates from one cell to another cell that happens to share the same extracellular space. An example of this would be the inflammation response, and also how cancer cells survive. So in this example, you would have a signaling cell here that would produce several signaling molecules, and they would be, of course, released. But they don't enter a circulatory system like we saw with the endocrine system. They just enter this extracellular space. And you will have various target cells that share the extracellular space that will bind to this signaling molecule, providing some kind of response in these cells. This allows cells within a similar tissue or in a similar environment to communicate with each other so they can respond in a similar manner. Alright, so let's talk about our third one, synaptic. It's similar to the endocrine in that it delivers signal over a long distance. But it's different because the signal that is transmitted isn't spread as globally. Remember, the endocrine, the signal enters the circulatory system and can travel throughout the body. The signal within the synaptic system goes in one direction to one target. It's a process where an electrical signal, or I'll call it an impulse, is converted into a chemical signal. This occurs in specialized cells called neurons. So we'll have this neuron here that can travel greater than 1 meter to a target cell. And it could be a muscle cell, so we'll use that as our example. So what happens is we will have a signal. It could be a signal from a neighboring neuron, or it could be a signal from another cell. But it will signal the neuron that will generate this electrical impulse down the neuron. It will then release a chemical. So this is what I mean by electrical impulse to a chemical signal. So it will release small chemicals known as neurotransmitters. And it'll be released in this area known as the synapse. These neurotransmitters can interact with a target cell. It could be a specialized cell, it could be another neuron, but it will interact with a target cell to give a specific response. Okay, let's talk about the fourth communication pathway, and this was the contact dependent. So this one requires a signal molecule that is not secreted, but rather on the cell surface. So this signaling cell then binds directly to the target cell. As an example, this is really important in embryonic cell differentiation, where one cell will bind to another cell to stimulate a cellular change. So here we will have our signaling cell that has a signaling molecule attached to it, and it will bind directly to a target cell, which will lead to a response. Next, I'm going to spend a little bit of time talking about receptors and signals and how they interact. So remember, on any given cell, say a human cell or a plant cell, there are going to be many kinds of receptors. So a cell could have hundreds of receptors. When each one is bound, a different response occurs. And so it's important that only the right signal binds to the right receptor, and that the cell is able to differentiate between these signals. In addition, they have to balance within the cell the different responses. So each one of these will give a specific response, but each one of these could be activated at the exact same time within the cell, and the cell has to be able to balance the resources and work together with those responses. And sometimes the responses are coupled, going towards a similar response. So the signals are also going to be very specific to the receptors to achieve that process. So just as we talked about enzymes and ligands and the specificity, this is also an example of ligand specificity. So even though I drew these receptors on the outside of the cell, we can classify receptors based upon their location. They can be on the cell surface, as I've drawn here, or they could be intracellular receptors. They will respond to different kinds of signals. So we can think of signaling locations. So those that bind to the cell surface are more likely to be large and hydrophilic. they're not going to easily diffuse across the membrane. Counter to that, the signals that bind to intracellular receptors are more likely to be small and hydrophobic. They could be larger and hydrophobic, but they would definitely have to be hydrophobic because they would have to get across the membrane. And to give equal playtime, we should talk about these signals that come into the cell. So you could have a hydrophobic signal here that enters into the cell directly and then binds to, let's say we have the nucleus here. It may bind directly to a receptor on the nucleus to give a desired response. It could also bind to a protein here. I'll just put P here. A protein here that then binds to this nuclear receptor to cause a response. But at some level, this intracellular signal enters the cell and delivers the desired response. Next, I want to talk about the variable speeds of responses. So we will have our cell here, and let's draw two receptors. Now each of these receptors are going to bind to and respond to a different signal. Here I drew a square signal, and here I drew a triangle signal. Over here I'm going to show a slow response, and over here I will show a fast response. Sometimes when a signal binds to the receptor, sometimes what it needs to do is to modify a protein that's already made. So I'll just make it very simple here, from a square to a circle protein. But this new one here is the active protein. And this active protein overall changes the cytoplasmic machinery, meaning it's changing this protein. Maybe this signal targets the nucleus. Maybe the DNA in here is triggered to make a messenger RNA. So now we have a messenger RNA. This messenger RNA is taken out of the cell, and once in the cytoplasm, it is translated into a protein. This also, just like the fast response, leads to a change in the cytoplasmic machinery. A new protein has been made. Both of these will, after changing that machinery, probably not the same pathway, but it could be, a change in cell behavior. That behavior could be to divide, it could be to change shape, it could be to develop, it could be to die, it could be lots of different things. But you can see here how this is going to be much faster, because this signal directly changes a protein, and now it can change the overall machinery of the cell. Here you have to enter into the nucleus, make the messenger RNA. The messenger RNA then is transported out of the nucleus and is converted into a protein. Many more steps here, many more time-consuming steps that must occur before we can change the overall cytoplasmic protein machinery. They both lead to a cell behavior change, but this just takes longer. An example over here for fast would be muscle contraction. This can occur in 1 millisecond. So a very fast response. Over here is an example of something slow, would be cell division. This can take many hours. Depending on the organism you're talking about, it can take upwards of 18 to 24 hours to copy that cell. Now in both of these cases here, I've drawn this, and in other slides I've drawn this kind of weird little squiggly line. And I've called it intracellular signaling. But I haven't really explained what's happening here, or what could be happening here. So I think it's important to talk about this mysterious squiggly line here. So for simplicity, I'm just going to use this receptor here. When this signal binds to this receptor, several things can happen. But let's just say it changes the overall shape of this receptor. And now it has just a change there. This change can now signal another protein, which can then signal another protein, another protein, another protein, and it can go on and on. But ultimately, at some point, what will happen is this protein here, at the end of it, will ultimately alter an effector protein. Maybe it will change the shape or activate a cytoskeletal protein, which will give you a response of, say, cell shape change. Maybe this effector protein is an enzyme, and that's going to change the cell's metabolism. Maybe this effector protein is a transcription regulator, and that's going to change gene expression. So we have a signal, it binds to the receptor causes this chain of events of signaling downstream molecules to ultimately change an effector protein, and depending on what that effector protein is, it can have variable responses. So what I want to say here next is answer this question of why. Why do we have to have this long signal here? Wouldn't it be easier if we just signal this molecule here and this molecule immediately altered the cytoskeletal protein, an enzyme, or a transcription factor? Well, the answer is no, because otherwise I wouldn't be talking about it. But why is the answer no? Having multiple steps here aids in regulation. You don't move down this pathway unless the previous pathway has been activated. You can also have one signal with multiple responses. So maybe there's a complex response that's needed, and we need to change the shape, we need to change metabolism, and we need to change gene expression. Maybe this molecule can affect all three. The last one I want to talk about here is called amplification of the signal. I didn't draw it that way at first, but we can do it now. What happens if we have 3 of these molecules? And what happens if we have 4 of these? And what happens if down here we have 100 of the triangles? And maybe 1,000 of these circle proteins. This one signal can activate these 3, which then can activate many, many more of these, and down the line. Just one signal can ultimately make this final protein here amplified because we have many more of these proteins. This can allow for a much quicker response and a much stronger response. The next thing I want to talk about is how cell signaling can act as a molecular switch. How the cell can turn on or off a pathway simply through a signaling pathway. And I want to talk about this using a protein phosphorylation event and also a GTP hydrolysis event. But let's talk about phosphorylation first. So up here will be the off state, and down here will be the on state. A signal from outside the cell, don't know what it is, doesn't matter right now, activates a protein kinase. Activating this protein kinase will add a phosphate group to this protein. This now activates this protein and this switches on. And now we get some kind of response. But if we want to turn off that response, then a protein phosphatase will remove that phosphate group from the protein and turns it off. Signal comes in, activates kinase, puts a phosphate group on the protein, the target protein, to get a response. We turn it off by removing the protein using a protein phosphatase. So to remind you, this was a protein phosphorylation example. Now over here, let's look at a GTP hydrolysis example. Again, we'll have our off protein up here and our on protein down here. We'll get a signal from outside the cell, and it will turn that protein from an off state or an inactive state to an on or active state. And it'll do so by activating this GTP transfer protein, where it will exchange the GTP found on this one with GTP. So now it has GTP on, it's active, it's on, and we will get a desired response. But we don't always want that response on, so there will be a time when we want to turn it off, and a GTPase will be activated to hydrolyze this GTP, leaving only GDP bound to it in its active or off state. So again, signal comes in, activates this GTP transfer protein, replaces the GDP with a GTP. Now it's on, and we get a response. To turn it off, the GTPase hydrolyzes GTP to leave only GDP on there to turn it off. So fairly quick and efficient way to turn a system on or off to respond to a specific stimulus. Now the last thing I want to talk about are cell surface receptors, types. We've been talking about them a lot by just drawing them as generic receptors that bind to some kind of signal that gives a response. And there are three kinds of receptors I want to talk about. The first are ion channel coupled receptors. The second are G-protein coupled receptors. And the third are enzyme coupled receptors. So I want to introduce these three. In the next series of podcasts, we'll talk more specifically about G-protein coupled receptors. And to close off cell signaling, we'll talk about enzyme coupled receptors. But let me introduce all three of them first. So let's first talk about ion channel receptors. So let's draw a membrane here with a receptor here. And let's say we have an ion, and it could be calcium. And it cannot get through this receptor. It's blocked. But what can happen is this receptor can bind to a signal. And that signal changes, it could change the charge, it could change the conformation. But ultimately what happens is this signal-bound receptor will allow the ion to now go through. Once in here, this ion, or calcium specifically here, can have several downstream effects. And we'll talk about some of those effects later. But this is how an ion channel coupled receptor would allow an ion to get into the cell in response to a specific signal. Next, let's talk about the G-protein coupled receptor, an infinitely important receptor complex. And we'll spend quite a bit of time talking about this on the next podcast. But let's draw our membrane here. All right, so let's begin by drawing three different protein components that are important in the G-protein coupled response. We have our receptor here. We will then have what's known as a G-protein, and then we will have a enzyme here. And as drawn here, this would be inactive. Well, how do we activate it? Well, it's activated by the receptor, probably not horribly shocking, binds to a signal. And when it binds to this signal, it will activate the G-protein. Once it's active, the G-protein can now activate the enzyme. This interaction between the active G-protein and the now active enzyme is critical in order to get the response. Of course, it will involve an intercellular signaling pathway before you get to the response, but ultimately this is the first part of the intracellular pathway of having these two protein complexes interact with each other. Okay, next let's talk about our last one, the enzyme coupled receptors. Now these can be one of two types. The first one is when the enzyme is the receptor. So in this case, we will have our membrane shown here, and we will have our two receptors. Right now they're inactive. But when a signal comes along and binds to them, it will facilitate their interactions with each other. This now leads to an active receptor enzyme, which of course will lead to an intracellular signaling pathway and ultimately to a desired response by the cell. Alternatively, the enzyme interacts directly with receptor. In this case, you have your plasma membrane. We'll draw our two receptors that are inactive. The signaling molecule, just like before, will bind to them in such a way that allows them to interact. Now, in this case, they are not enzymes themselves, but what they will do is interact with this previously inactive enzyme and activate the enzyme. And then this will go on to stimulate an intracellular signaling pathway and give you that desired response for the cell. Okay, that's all I have for this podcast. If you have any questions over the material in this podcast, please let me know and I'll help you through it. If not, I'll see you in class. Bye.